Thank you for tuning in today to hear about the work we've been doing analyzing the various types of data that are being collected in Answer ALS. As you've already heard, Answer ALS has collected many different types of data. There are genomic data that are collected directly from the patient. There are data that are derived from induced pluripotent stem cells that are grown in the lab into neurons. And from that, we get epigenomic, transcriptomic, proteomic data. And then there's a lot of clinical data that we gather as well from the clinicians from the medical sites. And lastly, you'll be hearing a little bit about the app, the phone app that's being used to monitor behavioral changes in ALS. And our overarching goal in this project is to provide the data to the entire public, scientific and lay audiences, so that everybody can have an opportunity to try to dive into the data and discover if there are different subtypes of ALS. Because we believe fundamentally that ALS is not a single disease. And just like cancer cannot be cured by a single pill, there will not be a single treatment for ALS, but hopefully we'll find the subtypes of ALS, and each one of those will have associated with it its own set of therapies. A lot of these data are already available. You'll hear more about the portal that we've created as well, but it's there right now for you to analyze, data.answerals.org, and I encourage you to go there and take a look. With all of these different kinds of data, we've also been very busy internally in Answer ALS trying to understand whether there are different subtypes of the disease. And I'll talk quite a bit about the clinical features of the disease in this talk. In particular, we'll be using something called the ALS Functional Rating Scale, or ALS-FRSR. This is a 48-point scale that measures symptoms associated with the disease. A 48-point score on this scale means that the patient has no symptoms, and then as the disease progresses, the rating scale declines. And it consists of four main types of questions that are asked of the patient that relate to bulbar function, function around the throat, speech, swallowing, fine motor skills, gross motor skills, and then breathing. We started out with a very simple question: are there different typical patterns of progression in ALS by using this ALS functional rating scale, monitoring how it changes over time, and then looking for patterns? Are there clusters of patients who all show one behavior or another? And Divya Ravamuthi from my lab teamed up with a group at IBM Watson to explore this question in detail. The real data look something like this. So this is a fairly large sample of data from Answer ALS, where each patient, when they responded to the questions on the ALS functional rating scale, was given a dot. The lines connect the data for a single patient. And you can see it's very hard to discern any global patterns in such a complex data set. That's all the more the case because we actually observe each patient for a very short time, typically about a year. And so during that period of a year, we might have 2 or 3 measurements of the patient. And because this is a rating scale that's measured by asking the patient questions, you can imagine that their responses might differ from day to day. Maybe one day they're feeling a little bit better and they do better in their walking, and the next day they're feeling worse. And so there's inherent noise in the measurements. So we have sparse data, we have noisy data, and then we've got these we have a challenge to assemble these patterns from all the sparse and noisy data. So we, together with our IBM friends, established an artificial intelligence-based approach for doing this, something called a mixture of Gaussian process model. And this model allowed us to identify certain common patterns, which are designated by the dark lines here, and the spread, the variance that we would expect to see around each of those patterns. And in analyzing the Answer ALS data, we can then discover a couple of common patterns that patients take through the course of their disease. I'll show you some results from a very large clinical database outside of Answer ALS called PROACT, where we applied the same model. And in these pictures, you'll see black dots that represent the clinical data for each patient. The blue lines and the shaded region around those blue lines represent our AI-based model. And the dashed lines will represent a basic model that's commonly used in the disease analysis that I'll discuss in a moment. And so when we analyze these large databases using a mixture of Gaussian process model, this artificial intelligence-based model, we pick up several standard patterns that can be seen to be quite reproducible. There are patients, as shown here, who have fairly stable linear decline in their disease over the course of time. There are others who show a concave or convex, a curved shape to the trajectory that the patient takes through the course of the disease. And most interestingly are these patients who have what's called a sigmoidal curve, an S-shaped curve. where initially the disease seems stable, slowly declining, then it undergoes a period of rapid descent, and then it slows down again. Importantly, the patterns that we detect in one data set are transferable to another. So here we've trained our model on the PROACT data set, and here's one of the many patterns that emerged. And now we can superimpose on this PROACT data, the pattern from the PROACT data, the actual data from ALS, the complicated data set that I showed you earlier. And we can see that amongst all of those patients in ALS, there are a group indicated in green who have the same pattern that was observed in the PROACT data set. This is going to be incredibly important because it allows us to then say that these are probably not just random features of the patients, because they're reproducible from cohort to cohort, from groups of patients studied at completely different times, different clinicians, different medical institutions. And we believe that in analyzing these and trying to get at the molecular basis for these subtypes could be critical to advancing the search for therapeutics. It's not only the overall ALS-FRSR that indicates these patterns of decline, but we can also detect it in the different types of questions that are asked. So I mentioned before that it's composed of 4 different categories. And here's an example showing that the bulbar category that measures speech and swallowing and other things like that also shows some patients who show very little decline with time and others who undergo rapid descent. There are other clinical measures that are not part of the ALS-FRSR, but because we have such a rich database of clinical data in ALS, we can examine those as well. And one of these is forced vital capacity, or FVC, which is a measure of breathing capacity. And here too we see quite a variety of patterns that are experienced by patients as they go through the course of the disease. The fact that not all patients undergo a linear decline in their disease actually has important implications, because the vast majority of modeling in the disease has been focused on these linear patterns of decline. And in fact, many clinical trials determine whether or not a drug succeeds by how much the patient's progression differs from that linear decline. Here are some results from a real clinical trial that was judged to have failed. It was called ceftriaxone. And we can see that several of the patients, 68 different patients shown here, match patterns that we've detected in other databases of disease, ALS and PROACT. Now, if you applied the standard clinical modeling of the disease, you actually would have come up with very wrong predictions for these patients. In some cases, the standard modeling would overestimate the patient's decline, and in other cases, it would underestimate their decline. If you're misestimating what's going to happen to the patients in the absence of the disease, you can understand why it's so challenging then to predict which patients are benefiting. And it's possible that some of these previous clinical trials that were deemed to have failed would have actually succeeded if we had been able to separately study the results for different subtypes of the disease, different clinical patterns of decline. So far, I've told you about our work focusing on the clinical subtypes of the disease. But of course, ALS has a very, very rich database of molecular data. And we're critically interested in whether there are relationships between the clinical subtypes and the molecular subtypes. We know in principle that this is possible, so it's been previously shown that certain variants of genes do associate with particular clinical patterns. For example, there's a gene called SOD1 or SOD1, and one of the variants that's seen in ALS patients called A4V is associated with very, very steep decline. Similarly, it's been seen by a number of studies that patients with variations in the C9orf72 gene tend to decline more rapidly than other patients, but it's by no means a hard and fast rule. And so it's conceivable then for us to be able to find different molecular features, maybe not all genomic variants as shown here, but perhaps changes in proteins or the epigenome that might also be predictive of clinical behavior. But there's a challenge here, because it's very easy to find variations in the genome, the epigenome, the proteome that we can find differ across ALS patients and to group them into different clusters. But because there are over 3 billion bases in the genome and hundreds of thousands of proteins and genes and epigenomic regions, there's a chance that all of these variations are occurring just by chance. Just like, if 2 people win the lottery on the same day, it's not because there was something inherent in those 2 people that made them win it. It was just luck. And so if 2 ALS patients share a certain pattern or variation in genes, it could be that it represents a subtype of disease. But it's also very, very likely that it's just luck that they have those common variations. So how do we tell the difference? Well, one of the things we're doing is using a kind of computational approach, an AI-based approach called causal modeling. And we have on our ALS team Karen Sachs, who's an expert in this area, and she's been developing some specific approaches that could be tailored to the ALS study. And we've also teamed up with a company that focuses on this called GNS Healthcare. And together we're working to analyze all the different kinds of data and try to identify those variations that are actually causing the disease, causing phenotypes, molecular changes that are related to what the patient actually experiences, and distinguish those from the molecular changes that are present in those patients but just present by chance. There's another approach to this problem as well, and this is being spearheaded by Johnny Lee, a graduate student in my lab. And here I'll show you a proof of concept for how we can distinguish different types of molecular changes in cells. Here we focused on patients who all had a common, here we focused on patients who all have the same genetic variation, the C9orf72 variation in their genes. And using iPS cells, just like in the full project, we focused on molecular changes that can be detected in their iPS cells. So we gathered a lot of data, and then we used a modeling approach that we've developed called Omics Integrator to identify patterns of molecular changes that seem to be coordinated. You might consider these pathways through the cell where a molecular signal gets propagated from the gene and then on to different parts of the cell, onward and onward, until it leads to something measurable or something experienced by the patient. Now, these patterns could be part of the cause of the disease. They could be random chance patterns that are not particularly relevant to the disease, but also through Johnny's work, we have identified another kind of causality, which we think is very important. So how do you tell whether a molecular change is relevant to the disease or not? Well, the best thing to do is to go back into the cells, change some of those molecules, and see how the cell responds. It's quite expensive and time-consuming to do that in iPS cells, although we are gearing up to do that in the next phase of ALS. But for interests of speed, we focused on expressing the same gene that causes ALS, C9orf72, in a Drosophila, in a fruit fly, the kind of fly you might find flying around your house. These expression of this gene in the fruit fly causes loss of neurons, and particularly we looked at loss of neurons in the eye of the fly, which is very easy to measure. And then we manipulated individual genes that were identified by our Omics Integrator approach and asked what effect they had on the neurodegeneration. And through this process, we can go through an actual cycle of iterative refinement where we carry out measurements, modeling, and experimentation, manipulation, until we have a good model of what's going on in the cell. And this reveals something fascinating. So as I told you, all of these cells that we're looking at either have the C9orf72 variant that's present in the patients or they're control cells. And we can see patterns of changes in a set of genes that are involved in nuclear cytoplasmic transport. And we can tell through the fly analysis that this is part of what's actually causing the disease. It's making the phenotype, the neurodegeneration in the eye of the fly, and therefore in the patient as well, worse. But there are other molecular changes that have the opposite effect. They are directly caused by the C9orf72 variation. But these are part of the cell's natural response. These are stress response genes. And being able to distinguish between these genes that are contributing to the disease and these that are part of, and the ones that are part of the natural defense of the host against the disease is really critical. If we're thinking about what to make to target to make a drug, we want to prevent the changes that we observe in nuclear cytoplasmic transport, but increase the changes that we see in the stress response so the cell can fight back. And there are many genes and pathways that fall somewhere in the middle. So DNA repair both contributes in some cases and prevents the changes that are associated with neurodegeneration. And there are a whole host of pathways that Johnny has been able to map out, some of which are contributing to disease and some of which are compensating for the disease. So we believe that this type of causal modeling, both the computational approach and the mixture of computation and experiment, is really going to be critical. In the past year then, to summarize, we've done a lot to try to identify clinical subtypes of the disease. And we have a robust set of trajectories of how patients progress through the disease that we think can be quite useful both in understanding the ALS data and more broadly for clinical trials. We've generated an enormous database of molecular changes. And so we hope that in the coming year, we'll be able to better understand the associations between these clinical changes and the molecular changes. And in particular, we hope to identify causal paths that could be targeted therapeutically. With that, I'd like to close and thank all the members of the ALS team, especially the members of my lab who contributed to this research, and especially the patients and their families who participated in this study.